We're in the second section of genetics and behavior. So we have different types of genes, and I started talking about this to some extent with the chromosomes on that first slide. Pictures tell me a lot of what I want to say and need to say, but here we have the language on a slide. So we have autosomal genes. So all of those genes on the autosomal chromosomes. Then we have sex-linked genes, the genes that are located on those sex chromosomes, the X and the Y that I showed in that very first pictures. We were talking about Mendelian genetics. In mammals, the sex chromosomes are designated X and Y. Females have the two X's, males have an X and a Y. The two X's have are each showing the strands of genetic material. The Y has, if I remember correctly, something like 1/3 of the genetic material, and so there's no kind of matching part of the chromosome for about 2/3 of that other X chromosome in males. So during reproduction, because I'm a female and I have two X chromosomes, the female is going to contribute an X chromosome necessarily. Males can contribute either an X or a Y chromosome. Then that's going to determine the sex of the child. I usually do my brief. I cannot remember, I probably was my biology teacher in high school, so I'm sure you've heard this too, who mentioned, it's always one of the memorable things, how Henry VIII was upset that his wives weren't giving him male heirs. And so he would have he's probably upset about other things too, but he was having his wives killed when really that was him, right? Because he's the one who's determining the sex of the child. As I said, the human Y chromosome is small with relatively few genes of its own. And these are referred to as sex-linked genes, sometimes called X-linked genes, because it is the genes on those X chromosomes that are important here and that are different in males and females in that usually what we're looking at where we see those males as having a more likely phenotype of showing some trait is something that is a recessive gene. And so oftentimes we see the female has another gene that's the dominant gene, and so we're not seeing that genotype in the phenotype, in the observable characteristics. One example is red-green color deficiency. There are many examples, almost everything that I can think of that we see more as being more likely in males than in females. It's because it's on these sex-linked genes. Another example is hemophilia, and so that's what's in my picture over to the right. And I'm going to walk through the picture like we usually do, just in case you haven't done this a few times at biology classes. So if I'm the mother, I have, in this example, an X chromosome that's not carrying hemophilia and an X chromosome that is carrying hemophilia, and I'm just a carrier, right? Because again, the not having hemophilia is the dominant of that dominant recessive pair. And me and the father have a 50% chance, right, of having a male, 50% chance of having a female. So if we break this down, I'm going to start over here with this female who's not a carrier. If I, as the mother, give my X gene that's not carrying hemophilia, and the father's providing an X, we get a female who's not a carrier. If I give my gene that is the hemophilia carrying gene, and the father gives his X gene, then we have a daughter who is also a carrier. If I'm the mother and I'm giving my X that is not carrying hemophilia, and the father gives his Y, then we end up with a son who does not have hemophilia. If I'm the mother and I'm give my gene that is carrying hemophilia and the father gives his Y, then we end up with a son who does have hemophilia. Okay, and again, the daughter, to show hemophilia, she would have to have the mother having the gene that carries hemophilia and the father having hemophilia, because he would be giving his X that's carrying hemophilia, since it's a recessive gene. So much less likely. Your author distinguishes between sex-linked genes and sex-limited genes. And this is a really important distinction, that these sex-limited genes are genes that are present in both sexes. They're on the autosomal chromosomes, but they're activated by different hormones. And so we're seeing in the phenotype, we have different phenotypes, right? So mainly we're going to see the effect on one sex or the other. So genes for chest hair, genes determining breast size, and so forth. And genes change in several ways, which is going to be repeated very, very briefly when we talk about evolution, that we can have these mutations, which is a heritable change in a DNA molecule. And as we know, those mutations, usually they don't have much influence at all on my survival likelihood, but if they make it more likely that somebody in a species is going to survive and reproduce, then we're more likely to see that mutation being passed down to future generations. If the mutation makes it less likely for me to survive and reproduce, we're going to be less and less likely to see that being passed down in generations. Another form of gene change is these microduplications and microdeletions, which is a small part of the gene either being copied or appearing twice, maybe even more than twice, or being deleted out and not appearing at all. And one example is, I have schizophrenia here, but this is also what they believe for autism, that there's a genetic marker, but that part of what's important and part of what we're seeing in the genetic codes is that they're much more likely to have these microduplications and microdeletions of brain-relevant genes. Okay, and this brings us to epigenetics, which I used to have my students have a discussion on epigenetics. This brings a new kind of realization and understanding to the nature-nurture debate, right? Because we have these genetics, but we can see changes in gene expression, and those changes in gene expression are to the phenotype, right? This is not modification of the DNA sequence. I read a really great book probably 20 years ago, and of course I thought what he was saying made complete sense. It was like how to change your DNA. But as I've learned more and more, I've thought, well, that's a little bit a misnomer, because we're not changing our DNA, we're changing the expression of that DNA. Changes in genetic expression are important for such various experiences as how we handle stress, learning and memory, how the brain changes as a result from drug addiction. So we can see these changes because of our environment in which genes are more likely to be then transcribed into that messenger RNA, and which genes are less likely to be transcribed into that messenger RNA and so to be expressed. Epigenetic differences are likely the explanation for differences between those monozygotic twins. I mean, they're identical twins in their genetics, but they're going to have different environments. And as we're going to see, can't remember where exactly, if I do talk about it explicitly, so I will here, that even just handedness. So it looks like part of our handedness is because of the way we are lying in the womb, and most of us lie on our left side, and so our right hand is kind of free. But identical twins, they're next to each other, so it's likely that one is going to be right-handed and the other is going to be left-handed. And so that's happening in their environment, in prenatally. So epigenetics is about reversible genetic change. This is not a change in the DNA sequence itself, and I hope it becomes more clear how we're determining gene expression patterns through epigenetics, but when I start talking about histones, and it is influenced by environmental factors. And I'm going to give a couple of examples and hope that between the examples and talking about histones, this becomes kind of clear how important this is. So if I'm a parent and I'm realizing that I have either type 1 or type 2 alcoholism in my family and my genetic background, then I want to be very careful with my children. so that we don't see this genetic expression. I want to make sure that I'm not giving them an example of someone who's drinking too much. I want to make sure that I don't have alcohol around the house where they can get to it, especially when they're younger, especially when they're teenagers and likely to want to get into trouble. I want to communicate with my children about how important it is that they don't try alcohol, that they wait to try alcohol until their brain is fully developed, till after the age of 21. Now I'm more and more considering that maybe the age of 25, because that's when our prefrontal cortex is fully developed. And when they do drink, that they drink responsibly, that they're not binge drinking, that they maybe a glass of wine with dinner, that they're doing this kind of more safe drinking, if they drink at all, because then I know that this runs in my family. So what's happening is, again, they're not changing their DNA sequence with choices, but they are changing what's expressed of that DNA sequence. And we can kind of talk about how this happens with the histone modification in a way that I hope starts to make sense. So if you look at the picture, those round green-blue kind of looking balls, those are histones. And what this is showing is that DNA strand is wrapped around those histones. Okay, and if we have methyl groups that are associated with gene silencing, so that are making these, having the DNA be more tightly wound around the histones. And we have acetyl groups that are facilitating gene expression, so making it more likely that we unwind and expose a part of that DNA, and that exposed part of the DNA is more likely to be expressed, as you can kind of see in the picture. So, and it looks like it's not just about how my genetic expression, but it is also about what I'm likely to see in future generations, genetic expression as well, as there is some research showing that even how healthy my grandparents were can influence my life. And I can't remember the research, and I can't remember where I found it, but I think it's something about, if I remember, it's sort of about heart disease, but it might not be. Regardless, it's about health, and that we are passing this down. And I've seen one man talk about some longitudinal research that he did with his lab, where they were seeing whether or not there was alcoholism in the genetic background. They were controlling for that, so that they had a group of people who had no alcoholism in their genetic background as far as they knew, and they asked them questions about binge drinking, and then they looked longitudinally, and if they were binge drinking as teenagers, even if there wasn't this background of this genetic background for alcoholism, their children were more likely to binge drink as teenagers. So what we're passing on as far as this genetic expression is also important. I have one more example, just because I thought it was a really neat study, and it talks about this methylation, not so much about the acetyl groups, but about the methylation. So they looked at, this comes from Laura Freeberg's book, and I'm going to forget who the authors are. I don't think she gives them credit where I see it easily, but they looked at children who were maltreated, and those children and they looked at the likelihood of, so these sequences that are more likely to be methylated, and they had them compared to control children who had not been mistreated. And the maltreated children, so if we look at the high methylated, what is typically highly methylated, in control children, that's highly methylated, so it's more likely to be silenced. In maltreated children, it's less likely to be methylated, so it's more likely to be expressed. And they looked at what's low methylated, so what's more likely to be expressed. And that also was flipped from control children, so less likely to be expressed. The low methylated group more likely to be expressed. The low methylated group in control children, it was more likely to be silenced in children who are maltreated. Okay, I hope that all came out in the right order and it made sense, because I've looked at this a few times and it's really hard to talk through. But I think it's really important for showing how that kind of environment can influence our genetic expression and how it's happening through these histone modifications. And I think what the important point here from all of this is that what you do at any moment not only affects you now, but produces epigenetic effects that alter gene expressions for a longer period of time. So what you've done in the past influences you now and in the future, but you make choices now that are going to influence genetic expression, and not just for yourself, but for any children you may have. And potentially their children too, is what it looks like. Okay, so heredity and environment. Most behaviors and characteristics, as I've said, have both a genetic component and an environmental component. And this is why we talk about the nature versus nurture debate. If a characteristic depends largely on genetic differences, then it is said to have a high heritability. So I'm going to talk about this heritability rate and what factors are more likely to be inherited genetically. There are a number of ways that we get at these heritability rates, and oftentimes they're looking at concordance rates between different groups of people, and looking at the genetic similarity. A really common thing to do is to compare monozygotic twins and dizygotic twins to infer contributions of heredity and environment. So monozygotic twins being from one egg, they share their genetic background 100%. Dizygotic twins from two eggs, they are like regular siblings with about 50%, approximately 50% genetic similarity, but they are more similar in their environment than other siblings. They're more similar in their environments in the same way that monozygotic twins are more similar in their environments. Importantly, here is the prenatal environment, but what's also important is where you are in the family, as far as whether you're an older sibling, a middle sibling, or a younger sibling. Some of that has influence on who we become. So we have more similarity in environment between monozygotic twins and dizygotic twins than we do between other siblings. But we have this change in the percentage of shared genes. And I show here a picture of likelihood of schizophrenia. So identical twins are almost up to 50%. If I have an identical twin with schizophrenia, I'm 50% just about likely to be schizophrenic myself, and I'm going to jump down to the fraternal twin. If I have a fraternal twin, so a dizygotic twin, I'm down more at like 12 or 13%. And so it's that comparison of how much more likely are the identical twins to show the same trait than the fraternal twins. And this is a pretty high difference. And so we're going to say this appears to be pretty genetically driven. So one example of a twin study is looking at the heritability of depression and anxiety. If we're looking at 3-year-old twins, that heritability rate is estimated at 0.76, so very much looking like something that's inherited. By 12 years old, by 12-year-old identical twins, they are at a concordance rate of 0.48, so it's looking like environment has more and more of an influence and genetics less and less. And this makes sense. If we're showing attributes of depression and anxiety at 3 years old, that is likely because it's running in the family. And by the time we're 12 years old, environment has had more of a chance to have an influence, and we're more likely to be seeing effects of the environment. And this is, I keep this in here. I have cut out a lot of my heredity and environment, but I think there are some important points here. And this is an example of how environment never stops interacting with genetic predispositions. That we see these changes throughout our lifetimes. So another method for getting at this heritability estimate is to study adopted children and look at their resemblance to their biological parents to infer influences of heredity. So the logic being, if we're looking at an adopted child and they resemble their biological parents, even though they're not sharing that environment, then that appears to be very genetically driven. And if I'm looking at an adopted child and they look like their adoptive parents instead of their biological parents, then that appears to be very environmentally driven, since they're not sharing any genetics. So we look at the heritability of intelligence. Here, young adopted children are more similar to their adoptive parents than to biological parents in intelligence levels, but by the time they reach 16 years old, we have adopted adolescents looking more similar to their biological parents than to their adoptive parents. So again, we're seeing this interaction with environment that is occurring throughout our lives, right? That this changes from when we were very young to when we're teenagers. As to, at first it appears very environmental, and then it starts to appear more and more genetic. So going in the opposite direction of depression and anxiety, which makes a great deal of sense if you think about if I'm showing depression and anxiety very young, then that must be genetically inherited. So I have this kind of list of final points I want to make here, that heritability refers to how much characteristics are depending on genetic differences, and these heritability estimates are just estimates of how much is this due to genetics. And researchers have found evidence for heritability in almost every behavior that they have tested. And when we are looking at heritability of a certain trait, it is specific to a given population. And I'm going to show an example that I think makes it clear that we're looking at samples of a population, and the heritability estimate is from a sample to that population, and not necessarily to the population of humanity. I hope that makes sense. So, and then strong environmental influences can cause genetic influences to have less of an effect. So one of the things that they see with children who are adopted is that they tend to have a kind of similarity to their environmental experience, that they are usually adopted by people who are pretty motivated to have a child. They've jumped through a number of hoops. They have passed a number of tests. People have looked at them very closely. They usually have some money. And so there's not a lot of environmental variability. And if we put in this kind of environmental variability of, well, now we're looking at people who are wealthy versus people who are in poverty versus people who go to this kind of school versus people who are in a violent neighborhood, those larger changes make it look like environment has a larger influence. So our heritability estimates are going to be lower because of those large environmental changes and differences. Whereas when we have a more restricted environment, we're going to say, well, this appears to be very genetic. And so those are where these estimates are coming from. And I'm going to make the point one more time, I'm sure, because I know the slide that I'm talking about in my head, that we're looking at a sample, and it's referring to a specific population. So here's my example, and I think that I think it's important to digest this, so you know how important I think this is. That this came from this estimate of heritability of ADHD being among the highest comes from a number of samples. And we're going to compare it first to breast cancer, which has some genetic influence, but is very environmentally driven, as I'm down near 0.2 as the estimate. Asthma, again, it has some genetic influence, but environment is very important. Schizophrenia, we see that genetic predisposition is very important, but there's still an influence of environment. And height, of course, again, we see that genetic influence is very important, but there is an influence of environment. We're not going to see 1, right? We're not going to see a 1.0 where this is completely genetic. As I said, almost every behavior, we see an influence of both heredity and environment. And it's complicated to answer the question of how they are interacting, and we really need to look at this in several samples, several populations for every single behavior that we're asking about. So here's my estimate of ADHD, and it is up at 0.8. So between schizophrenia and height, it's among the highest of these kinds of disorders, or I like to call them differences in thinking. But what this is showing is this is a review study, and this review study is including that Hudziak study, Natter, Levy. If you look down the left side, those are different studies that each have their own estimates. Okay, and look at those estimates. Some of them are as low as 0.6, and one of them is really close to a 1.0, which is a hugely high estimate. So I'm making this point one more time, that those estimates are coming from samples that we are extrapolating to discuss about a population, and these are all based on twin studies, so they are samples of a particular group of people, meaning the twins. So I just think it's really important to keep in mind, because we take these heritability estimates as being sort of this important estimate, but really we have to keep in mind all the background of how they came to this estimate. And the more samples, the more different groups of people that we can look at, the better off we are at actually getting an estimate, an appropriate estimate for the larger population. And traits with even a very strong hereditary influence can be modified by environmental intervention. I know I read one of the student papers on this with, I think it was sickle cell anemia, but I'm going to give the example of phenylketonuria. So this is one of those, there's a dominant and a recessive gene, and I need to have the 2 recessive genes to have PKU, this phenylketonuria, which is the inability to metabolize the amino acid phenylalanine. So if you go look this up, if you're at all interested in this, this means there are so many things that you simply cannot eat, that you cannot metabolize, and if you eat enough of them, especially when you're young, but you can't even eat them when you're older, you're going to do some brain damage and some damage to your body. So if I'm getting these 2 recessive genes, I'm going to have PKU, but environmental interventions can modify that expression of PKU, of the phenylketonuria. I am going to have that regardless, but if we know that someone has it and we make sure they're not eating that amino acid in their food, the phenylalanine, they can live a relatively normal life. Whereas again, if they are in their environment, getting this amino acid in their food, which again, it's in so many foods that I don't know how they stand up on 2 feet, because it's in most proteins, and I'm a kind of a protein junkie because of my depression, that I feel like it's really important that I get my protein. But yeah, it's really their environment can make it so that they live a relatively normal life, or that they have some damage coming along. And I know I pull apart some of these points a bit more than Calit does, but I think they're really important points for our realization that genes are predispositions. They're not destinies. But my final point, and again, I think it's pretty important, is that genes do not directly produce behaviors. Genes produce proteins that increase the probability that a behavior will develop under certain circumstances. So in particular environments and with particular experiences. And genes can also have this indirect effect. We talk about a passive genotype to environment effect, or an evocative. The one I'm going to talk about here is an evocative genotype to environment effect, because genes can alter your environment by producing behaviors or traits that are going to alter how other people in your environment react to you and what they evoke basically from you. So genes can have various effects on how our environment influences us and influences that genetic expression.